Hello students, now we will start with new topic, that is Human Genome Project. What is aim of Human Genome Project? Its aim is, or its goal is, to study all the sequence or the genes, complete genes of human being, and use the accessibility of the genes and their functions for the next biological studies. Is to study about complete details of human gene sequence and use the accessibility of the genes for further biological studies. What is meant by genome here? Genome can be defined as the genetic material that is present inside one living cell. And also, genome is the genetic material of a haploid. DNA in a haploid chromosome is the DNA in a haploid chromosome. Means by taking one chromosome out of the pair and separating the DNA, that DNA we are giving a term like genome. The complete study of one chromosome can give us the detailed study of the remaining chromosomal gene sequence also. This Human Genome Project is a mega project. Why it is called as mega project? Because it was estimated that the base pair number is nearly 3 billions. Name is because the estimated base pairs in human genome is 3 billions. And also the cost of the study of each base pair, it was estimated as 9 billions. The cost to study each base pair, or one base pair we can tell. One base pair is 9 billions. Because of the cost, because of the study of number of base pairs, it was considered as a mega project. You will see some of the characteristic or the salient features of the Human Genome Project. Human genome, it contains how many number of base pairs? Means we said that it will be 3 billion in number. So coming to the millions of the nucleotide number, human genome contains approximately this number of, or 3.1 million number of nucleotides. And also average one human DNA has 3000 bases, nitrogen bases. In average, a human DNA, I'm not talking about the genome word here, human DNA has 3000 nitrogen bases. The largest human DNA or the gene, largest human gene has 2.4 million bases. Next, what is this largest human gene called? It is called as dystrophin gene. The largest human gene is called as dystrophin gene. The total number of genes found are only till now 30,000. Means we said that there are many number of base pairs, that is 3 billion, but the number of genes found are only 30,000 till now. The total number of genes that are discovered or studied or found till now are only 30,000. And among those, all the genes, 99.9% of the genes are of identical in all types of organisms. Function of 50% among these 30,000 genes are unknown. Next, less than 2% of genome means the total DNA in one haploid chromosome. Out of the total genome, less than 2% of the genome that codes for proteins. Now, by the time of 2003, all types of chromosomes are well studied, but chromosome 1 was not studied. Means Human Genome Project, it was started in October 1st, 1990 year and was stopped in or finished in April 14th, 2003 year. So totally Human Genome Project, initially it took 13 years for complete study. Nearly 1.4 millions of DNA locations means the nucleotide locations of DNA nucleotide locations are identified. They are given a term like single nucleotide polymorphism, termed as single nucleotide polymorphism. Now, what is meant by this single nucleotide polymorphism? This one is also termed as SNPs, SNIPs. What is this single nucleotide polymorphism? I previously told that 99.9% of genes are same in all organisms. Suppose think that there is a person A, person B, person. So we'll take two persons, that is enough. His body is having normal gene sequence that is common in all this 99.9% of gene sequence, which is equal to that of normal healthy living organism. So his body is having some gene sequence like this. This type of gene sequence is responsible for healthy condition of the person. Now, if any one particular nucleotide is different in a person and that small change of the nucleotide is responsible for the disease of the person or disorder or mutation in that person, then such type of gene sequences is considered as abnormal condition of the person. So, in an disordered person, or diseased person, there will be change in that one particular nucleotide sequence. Everything will be common, but the diseased person will be having change in one particular nucleotide. It can be cytosine or guanine or adenine or anything, or uracil. So this type of arrangement is called as single nucleotide polymorphism. So study of the arrangement of a normal nucleotide regularly which is in a person. If we study what is the location of so and so nucleotide, we can analyze what are the nucleotides or the genes that are responsible for the disorders, genetical disorders in the person. Or because of any types of diseases, if the person's genes have made some changes in their body, that also can be studied. This everything is possible only when the complete gene sequence up to the nucleotide level are perfectly known by the scientists. Human Genome Project was perfectly completed in 2006. Here we have written up to 2003, that is 13 years. But by the time chromosome 1 was not studied, but chromosome 1 was also finished its complete study by the year 2006. Was completed along with study of chromosome 1 in 2006. Means totally it took 16 years by the time of finishing of chromosome 1. Means chromosome 1 is studied between, means its complete successful study was done between 2003 and 2006 successfully. Before that also the studies were done, but the complete gene sequence was not clearly obtained. For this Human Genome Project, there was another type of biological study called as bioinformatics. The introduction of bioinformatics gave the support to Human Genome Project, and the bioinformatics was a simultaneous study with Human Genome Project. During the initial studies of this Human Genome Project, many ethical issues were raised. Means people who are objecting the Human Genome Projects, their version is we should not, means we don't have any right to disturb the nature and study about living organisms in the genetical way. But if we know that, then only we can give the best biological studies to the next generation. For that, this Human Genome Project was started in 1990 by objecting and crossing all these ethical issues and all that, and it was finished by 2006. This project was initiated by and perfectly studied or done under leadership of Collins. By US Department of Energy and National Institute of Health. This National Institute of Health is indicated with capital letters NIH. Next. What are the methodologies to study this Human Genome Project? Methodology to study is of two methods, means studying of the complete genome A to Z. Because in DNA we know that there are coding sequences and non-coding sequences. So study of the complete coding and non-coding sequences is termed as expressed sequence tags. So methodology is of two methods. One is studying the complete coding and non-coding sequences, which is termed as sequence annotations. Studying or sequencing of entire genome or entire DNA sequence is termed as sequence annotations. This is written with capital S and capital E. Second method is studying or sequencing only coding sequences. This is termed as expressed sequence tags. This is written with shortcut capital E, capital S and capital T. Expressed sequence tags. Now the question has to be arised. What is the need of studying non-coding sequences? What are non-coding sequences? So the question now has to be arised. What is or why should we read about non-coding sequences? First, we will see what are examples of non-coding sequences. In transcription, we have seen one term, that is introns. Introns are not coding for anything. So during the transcription method, in case of eukaryotes, the spliceosomes have been involved. Other examples are satellite DNA, next telomers, and VNTRs, variable number tandem repeats. So what is function of these non-coding sequences? They are not involved in transcription or coding for anything, but they involve in regulation of the genes. They does regulation of genes, but not involved in transcription and translation. So they are called as, because of lack of these two functions, they are called as non-coding sequences. For this Human Genome Project, first of all, if a gene sequence or a DNA, if it has to be studied, it has to be first cut in presence of endonucleases. We need a host for entering of the DNA into the host, and also for transportation. For this project, a vector is also needed. Mostly two types of vectors are involved here in this Human Genome Project. So vectors used are bacterial artificial chromosome and yeast artificial chromosome. This is yeast artificial chromosome. Among these both, yeast artificial chromosome is considered as the largest vector that is used for Human Genome Project. Among these both, which is the biggest one or largest one means yeast artificial chromosome vector is considered as the largest or biggest type of vector in Human Genome Project. In this Human Genome Project, when the sequences in the DNA has to be studied, first they will be incorporated into the host cell with the help of bacterial artificial chromosome or yeast artificial chromosome as vectors. So DNA in the host cell, they get replicated. Then after obtaining of sufficient number of DNA fragments, then they are taken out and their gene sequence is studied with the help of Sanger's method. When DNA in host cells are replicated and after obtaining of sufficient DNA fragments, then the sequence of nucleotides or genes is studied by Sanger's method. What is this Sanger's method? In case of Sanger's method, first step is denaturation of double-stranded DNA. Means the DNA which is double-stranded when heat is applied by applying heat. Then the strands become unwinded. Initially, they will be very nearby like this. Now, the distance has been increased because of application of the heat. Now, they are taken into reaction vessels or the test tubes. Vessels or test tubes. And now primers are added. Now each strand will behave as a template separately. Each strand behaves as a template. After taking into the reaction vessel, primers are added. After addition of the primers, then DNA polymerase is also added. After addition of the primers, then the total DNA plus the primer, according to the sequence, it is equally distributed into 4 different test tubes. The total content is equally distributed into 4 different reaction vessels or the test tubes. Now, the diagrammatic representation we can give like 1, 2, 3, 4. After this, DNA polymerase was added. DNA polymerase was added in all. So here we get the DNA sample along with the primers. What are present here? DNA plus primer. To this DNA plus primer, now we are adding the third substance, that is DNA polymerase. We can label this one as, this is containing DNTPs, all these 4 test tubes. This is A, B, C and D. What are DNTPs? DNTPs are deoxynucleotide triphosphates. For suppose think that these are all having DNTPs taken in 4 different test tubes. Full form is deoxynucleotide triphosphates. To this, all 4 number of DNTP reaction vessels, especially modified DNTPs are added. What are these DNTPs? These are termed as deoxynucleotide triphosphates. Then what is difference between DNTPs and DNTPs? We know that a nucleotide is having pentose sugar, having nitrogen base, carbon 1, 2, 3, 4, 5 and phosphates like this, a nucleotide triphosphate. At second carbon there is hydrogen for deoxyribose sugar, and at third carbon there is OH group. If OH group is present, then that can be called as deoxynucleotide triphosphate. If the oxygen is removed and only hydrogen is present, then that type of nucleotide is called as dideoxynucleotide. That is specially modified for sequencing of this one. Now, are added again to 4 different test tubes that we have taken here. So here DNTPs are already there. We have already added the RNA primers. We already added DNA polymerase. To this, we are now adding the modified deoxynucleotide triphosphates. For suppose, think that this nucleotide triphosphate is having the nitrogen base adenine. Here DNTPs are added, it is having guanine. Here also DNTPs are added. Here the nitrogen base is cytosine. Here also DNTPs are added. Here the nitrogen base is thymine. 4 different types of nitrogen bases are specially modified in the DNTPs and added to the 4 reaction vessels. This is A, B, C and D. Now, all these are mixed and allowed to wells of polyacrylamide gel electrophoresis. This polyacrylamide gel electrophoresis is considered to be better than normal agar gel electrophoresis because of its more resolution capacity. Is taken better than agar gel electrophoresis due to its high resolution power. The next step is now the DNA strands will get separated basing on the length of the DNA strands. Means the shorter DNA sequences or the shorter DNA strands, they will be moving far from the wells. Now the polyacrylamide gel electrophoresis. Here DNA are having negative charge and a electrode is connected to this. The DNA fragments, they tend to move towards the positive direction or positive charged area from negative to positive. Here DNA is having negative because of the phosphate ions. Because of phosphate ions in the DNA, it attains the negative charge. Now when it is allowed in the this polyacrylamide gel electrophoresis by supplying the current or the electrodes which are having the power connection, then the smaller DNA fragments will be moving the longer distance. Same like that of agar gel electrophoresis. Now, the sequence of this longer or distant DNA sequences, they are studied first, and the study is from the longer to the sequence of the DNA which are near to the wells. For suppose we will take few sequences and how the total gene sequence of the DNA is studied. Suppose think that here the longer DNA sequence are having adenine, guanine, thymine, cytosine sequence. So the next one which are near to this will be having adenine, guanine, thymine, cytosine, because these are not that much shorter than the first one. So it will be having another base pair. Again, the third one, adenine, guanine, thymine, cytosine, thymine, it will be having another nitrogen base pair extra. So this one, adenine, guanine, thymine, cytosine, thymine, adenine, one more will be there. It can be any of the base pairs. Another sequence, this one, fourth one, adenine, guanine, thymine, cytosine, thymine, adenine, cytosine. The last base pair can be any of the 4 nitrogen bases. Now we will take this one, adenine, guanine, thymine, cytosine, adenine, and it can be adenine. The last but one, adenine, guanine, thymine, cytosine, thymine, adenine, cytosine, guanine and adenine. Last can be thymine. The first one which are very near to the wells, adenine, guanine, thymine, cytosine, thymine, adenine, cytosine, guanine, adenine, thymine, and the last can be any of the nitrogen base. Now if we want to sort out what is the gene sequence here, we can write the gene sequence like of these edged. The gene sequence now can be written by taking adenine, guanine, thymine, cytosine, thymine, adenine. I am taking only the last base because all the same are equal. Cytosine, guanine, adenine, thymine, adenine. In this way, the sequence of the genes or the nucleotides or the nitrogen bases can be studied by using Sanger's method. Actually, Sanger's method complete study is not included in NCERT. I think it is not useful. But if a student gets a doubt, what is that sequence, how the method is done, means this will be the process. Next one. What are the applications of Human Genome Project? The last topic. Applications under Human Genome Project. The next one is DNA fingerprinting. So now we will see applications of Human Genome Project. What is use of this one? To diagnose a disease and give treatment. To diagnose a disease means basing on the type of the disease, the gene sequence can get varied, and for further treatment. Next one. If the gene sequence of a deceased person is known, then if possible, the treatment can be given by using rDNA technology. Treating disease or any disorder by using rDNA technology. Next one. Genome of non-humans can also be sequenced because we take some of the organisms like E. coli when we are doing the rDNA technology methods. So, we can study the gene sequence of non-human organisms like first one bacteria, second one Drosophila, third one rice and yeast also. So this information, we can use the information regarding bacteria, rice in the field of agriculture, and by taking the Drosophila and yeast, we can also study in the field of human further studies, genetical studies. So this information is used in field of agriculture and sufficient energy production. So this is about Human Genome Project and its applications.